everyone, and thank you so much for joining us today. I'm pleased to introduce my colleague, Jeanette May, our field application scientist at Countable Labs. Jeanette has been working with many of our early access customers in the rare molecule space to help them make the most out of Countable PCR and develop PCR-based assays that deliver high sensitivity results with faster turnaround times and less optimization. So Jeanette is going to share today how Countable PCR improves the workflows of rare molecule researchers and overcomes the limitations of other PCR methods. So if at any time while she's presenting you have a question, please go ahead and leave it in the Q&A box below. I will be back at the end of the presentation to kind of help her field the questions, and we'll try to get to as many of those as possible. And so with that, I will hand it off to you, Jeanette.
Awesome. Thanks, Steph. Hi, everyone. Thanks for joining us for the webinar today. My name is Jeanette, and I'm really excited to share with you our platform here at Countable Labs, and really how we are able to deliver a solution that can finally detect biomolecules with unrivaled sensitivity. I'll be discussing specifically how our platform, Countable PCR, overcomes the limitations of digital PCR and qPCR. Okay. So as we've looked around the genomic space, what we've realized is that there is a growing need for high sensitivity applications, such as detecting latent disease or detecting disease early. And with this growing need for high sensitivity applications, what we found is that the tools that are available or technology that are available to our customers is actually quite dated. So things like traditional PCR, it's a 30-year-old technology that is just not really equipped to address the needs of the field today. And with that, I'd like to share with you some early feedback that we heard from our customers in the field right now. So this particular quote that I'm sharing with you is a testimonial from a CLIA lab collaborator. They've been looking at rare cancer mutations in blood samples, and they're trying to look for it in like a really high sensitivity setting. And currently they're using digital PCR in their labs. To achieve the minimum sensitivity that they're looking for, they needed to combine the data from 3 separate wells, meaning that every blood sample was actually run in 3 separate reactions due to the limitation of their technology that they have in their labs. But in contrast, after doing a collaboration with them, they told us that our platform doesn't experience the same limitations as other legacy platforms do. So not only do you get better data, with lower background noise, but then you also get to combine reactions all into one well. So essentially, what you're getting is better data output with less labor and cost input. So usually when you're considering legacy platforms, there's a particular set of platforms that we all recognize to be instrumental in the life sciences space, right? So I'm sure everyone here knows that qPCR is a reliable workhorse. Every molecular biolab has access to one for general gene expression analysis. But the major limitation of qPCR, though, is that it's not really built for rare molecules. I mean, just in the way that you amplify and derive fluorescent signal to begin with, you can easily miss rare molecules. So rare targets can be missed when you're trying to amplify it from like a background of a lot more molecules. So in the practical sense, these qPCR platforms are not really sensitive enough to actually reliably capture, say, like 1 in 100 events, which is, in the grand scheme of numerical detection, not actually sensitive enough for the level of like rare molecule detection that a lot of researchers actually need. And then secondly, qPCR by nature is a relative quantification strategy, meaning that you have to make and use standard curves. So you're not really directly quantitating your sample ever. And then third, multiplexing on these systems usually is prohibitively slow and difficult to optimize. So many researchers actually choose to forego multiplexing altogether rather than struggling to make it work in qPCR. So more recently, in the last 10 to 15 years or so, another technology came about called digital PCR or dPCR. And it really set out to solve some of the issues that I just talked about in qPCR, but in that, it actually created some more problems. So for digital PCR, it's still really hard to do rare molecule detection because of several limitations that exist on the platform. So if you think back to the quote that I shared from our collaborator a couple slides ago, they said that they had to spread their sample across multiple reactions to actually get the sensitivity that they required for detection. a big limitation of the dPCR platform or the dPCR platforms that exist today are that all the current instruments rely on microfluidics. So whenever your sample is handled with any sort of microfluidics, you actually lose a lot of your samples or your sample and your molecules inside of the chip itself, meaning that you have a dead volume where you end up discarding a lot of the signal even before it goes into your detection mode. So you're basically losing all of those rare molecule signals. And secondly, with microfluidics, you can't really load a lot of your reaction into or your sample into a reaction. So what we often see is that, let's say you start with like a 50 microliter sample. If the reaction volume in dPCR is only 10 microliters, it really forces you to spread your sample into multiple wells if you want to run your entire sample, making rare molecule detection that much more difficult and that much more labor intensive. And then lastly, with multiplex assays, while they're more achievable in dPCR than they are in qPCR, you still have to design and pay for primers and probes, which honestly take a long time to order and receive, and then still require extensive optimization. And that usually just makes this type of assay cost prohibitive for a lot of applications. So then this is where our solution delivers. We have a platform that overcomes some of the biggest limitations in PCR today. Here in the slide, I'm going to show how the Countable PCR platform really achieves this leap in quantitation. All the data I'm going to show here in this slide was generated using the same DNA dilutions to really showcase the variability from platform to platform when you're thinking about how to choose a platform to use for your application. So for qPCR, we all know it as a workhorse tool, but the issue is that it always is just relative quantification. So when you have DNA that you load, and as you increase the amount of DNA, you probably would expect a linear increase. But due to how qPCR works by nature, without a standard curve, you don't actually see the accuracy that you want. So while it is low cost per sample, you need to do the standard curve to actually achieve that kind of accuracy that you want, which means you have to put more money and more labor into getting that accuracy. When we look at the same samples using digital PCR, in this case, it's actually droplet digital PCR, so ddPCR. What you see is that ddPCR solves some of these problems. So namely, here we're estimating a rough number of molecules in the sample. You see that the line is straight, unlike qPCR. So you don't need to do a standard curve. But the key limitation of digital PCR or ddPCR is that the dynamic range is always really low. So when you get to a target amount where it's close to that upper range, you start to get pretty inconsistent results. And then when you completely blow out of that range, you just get completely inaccurate counts. But with our platform, Countable PCR, we solve these issues by basically providing the best of both worlds. So using the same DNA samples, you see that we get a wide dynamic range quantitation that doesn't really require a standard curve. Plus, you're getting it with great precision as well. So, and what I mean by like great precision here is that when you look at these replicates, so each of these purple dots is actually 4 different replicates. And what you're seeing is extremely tight error bars, indicating really, really good reproducibility. Where Countable PCR really delivers in the realm of rare molecule detection is for those wanting to achieve the highest sensitivity possible, basically. So our philosophy here is to never waste time and material to try to get to these endeavors. So as I showed, we provide high precision capability across a wide dynamic range, which means it's possible to precisely quantify rare and abundant targets all together in one run. And also, with our technology, you're able to analyze your entire sample all at once with no lost or dead volume, unlike other legacy platforms. 4, with a dynamic range and all these other features that I mentioned, you no longer have to split your sample into like multiple reactions, and instead you're able to just run each sample all in a single reaction. And then finally, designing assays can often be really hard for many, many applications, but our platform provides a straightforward way to multiplex, and our multiplex experiments are possible to design and optimize in days instead of months, which is really appealing to me. Now, I'd like to share with you some examples on how Countable PCR really brings its versatility into the rare molecule detection space. So in the past, rare molecule detection has really kind of been a brute force approach, where with other platforms or assays, there's often only one way to do it, and you just have to try to find the most sensitive platform and assay design. But with our approach, we actually have multiple ways to achieve that same goal. So the first of all, let's start by addressing what I mean when I talk about sensitivity. Here I'm showing you, relative to other platforms, how we are achieving many, many times better sensitivity, even versus the best current digital PCR platform. But how do we do that, right? Here's an example of an oncology assay looking for cancer mutations. So with our ability to load 3 micrograms of DNA per sample, we're simply expanding, well, we're actually vastly expanding the haystack in search of the needle that we're looking for. And with that amount of DNA, it's really evident how we are able to like stretch our dynamic range. We're able to count rare mutations down to 40 in 1 million background wild type molecules. And this is all in the same tube. And even at this super, super low range, we're still comfortably above the blank or background for this experiment. So in the previous slide, the previous example, I showed you a way to detect rare molecules when you have a lot of DNA. But what if, say, you have a really little amount of DNA that's maybe like diluted, such as like cell-free DNA or maybe cerebral spinal fluid, for example? So we can take a look at the same assay, but now just using diluted DNA. Another way of really improving rare molecule detection is to leverage Countable PCR's dynamic range. So in this experiment, what we did was we took a very diluted DNA sample with wild type and the JAK2 V16F mutation, and then did a pre-amplification, which is a 20-minute protocol, super easy. And then after this pre-amplification step, the tube will have these really, really low frequency JAK2 mutations now amplified. And that's also in the presence of the wild type molecules now also amplified. What you see here is that in an average of 3 or 4 replicates, is that before pre-amplification, you might not have a lot of material. So we're talking maybe the target is in like the single digits, but then like statistically, it's really difficult to report molecule numbers at that low end. But with the preamp, you've now boosted your rare signal to increase the statistical confidence. So you can think like law of large numbers and statistics. The rare signal as well as the wild type are now preamplified at the same time so that the mutant to wild type ratio is still preserved, but you're getting a much better LOD. In this case, I think it was about like 3 times better. So with Countable PCR, the broad dynamic range means that you can measure both the mutant and the wild type in the same tube at the same time without the need to dilute or split into multiple experiments. So the third way of thinking about this versatility for rare molecule detection goes back to my discussion around our collaborator who was combining multiple wells for each sample. So this is an example of a similar project that we did with an academic KOL. They're currently a qPCR user who are trying to detect a latent pathogen. So on this graph, each dot is equal to 1 animal infected with pathogen, and each point represents from 10 to up to 400 qPCR reactions. On the left side, where the pathogen is present in high amounts in the blood, they typically run about like 10 reactions. And then you see here, this dotted line shows the percentage of positive reactions. As you go lower towards latent pathogen levels in the blood, however, you start seeing lower sensitivity because it has such poor reproducibility. And then this is where the collaborator starts to overload, adding more and more DNA, more and more reactions, and more labor into trying to detect that latent pathogen. As it goes lower and lower in abundance, the pathogen at least, the qPCR actually starts to fail. So even when you're running hundreds of qPCR reactions, you're still not detecting it. On the other hand, for our collaboration with them, we took the same animal subject's whole blood DNA, and instead of running them from 10 up to 400 reactions per subject, we simply took each subject and ran each one in a single tube. So each bar is equal to one single tube here. And instead of having multiple reactions pooled, it's only one reaction. And so you can see across the animal subjects, we detect these latent pathogens in a single reaction, really showcasing our sensitivity, but really with like a fraction of the labor that was required in qPCR. So to bring it all around, how I think about rare molecule detection is that with this platform, you now have more flexibility in searching for your rare biomarkers. First is that you're able to just load a lot of DNA. You're more likely to find something when you're looking through a lot more of it. And second is that in cases that you have very little DNA, you're able to enhance the statistical confidence by doing a pre-amplification protocol. And then three, instead of wasting a lot of time and material running multiple reactions, you're able to consolidate your assays all into one single reaction. So I talked a lot about like all the benefits and the perks of how like what we can provide, but we can segue now into talking about how the system actually works. So how does Countable PCR actually achieve this kind of sensitivity that I'm bragging about? So allow me to first take a step back and really try to highlight the key differences in how platforms do their quantitation here. So on the left side, I'm showing you how the legacy platforms work. So qPCR here on the left is an analog signal that you have to draw a threshold for in order to determine relative quantification. And then for digital PCR, dPCR, it's digitizing some signal, whether it's in droplets or wells that have 0, 1, 2, 3 copies of DNA. You're digitizing that and then estimating the quantification. Keyword estimating. On the other hand, on the right side, how countable PCR works. The fundamental difference of this technology is that we're actually counting whatever molecules are in the tube. So that's why we call our platform Countable PCR. And then this is what the data looks like. Whatever single molecule you see in the tube, we're actually able to count it. So on the left side, I'm showing a 4-channel reaction that we've done and split into the individual channels. And each channel means one target. When you combine it all in one tube and you look closely, you actually see that the targets don't overlap with each other. You also see that each color is from one molecule. So whether it's a blue molecule, a green one, or an orange one, what we do in uncountable PCR is count the signal from each individual molecule and then give you that data directly. And the workflow to get those counts is actually really easy. So there's no like multi-day workflow or anything long turnaround times or anything like that, like previous technologies. The technology that we have is super powerful, yet is simple enough to actually just work in a single PCR tube. So I'll show you how we achieve this. For those of you who are familiar with DNA centrifugation columns, this technology is a mini spin column that goes inside of a PCR tube. You load your PCR mix into this spin column, and then you put that in a standard Eppendorf benchtop centrifuge. You spin it for 20 minutes. And what happens during that spin time is that the target molecules that you're looking for gets dispersed into the tube and then gets encapsulated in a unique matrix. So here is a blown up example of that matrix. Each of these little wavy line worm things represents a DNA target isolated by itself. The matrix generation generates millions of compartments per tube, and then the PCR mix, primerase probe, everything else gets dispersed into the matrix along with the targets. During amplification in a standard benchtop PCR thermocycler, the signal gets amplified into whichever target or color that you have specified. Because amplification happens by itself in a single molecule compartment, like shown here, the amplification of multiplexing assays in our platform is actually really straightforward and simple. So then detection and counting is also equally easy. You take that same PCR tube that you did the spin and the amp in, and you put it in our imager instrument, our benchtop imager instrument, and the imager scans the tube directly with hundreds of light sheets and then provides you with that true absolute count. So then that's the number you get, then that's your data. There's no standard curve needed, no statistical Poisson error correction or anything like that, unlike legacy platforms. So I can show you exactly what it looks like when a tube gets placed onto our instrument. So here, you're going to see a video of those light sheets that I just mentioned, moving through the z-axis of the tube. So each frame is one slice. There are 4 channels or colors available, and each channel is captured by the imager separately. We then have an algorithm that outputs the number of molecules for your targets, and this is all done automatically. So there's no like manual thresholding needed. And so when you come back to look at your data, it's already in its final form. So you're getting these counts here. You don't need to do any actual work on it. And also, so we have like, yeah, colors, and then we also, if you're looking to add more specificity into your experiment, say you have like a positive control or something like that of your sample, you can implement a optional built-in machine learning model to improve your sensitivity. So now that you know how this platform works, there's a ton of other features that I want to talk to you about that really enable this huge leap in what's possible for rare molecules. But I want to specifically talk about how we can provide you with the highest sensitivity for capturing the rarest molecules. So here I'm showing you an example of one slice or light sheet from the middle of a PCR tube across a broad dynamic range of molecules. So it's easy to see when you have like, whether you have like a low number of molecules or a really high number of molecules, from tens to like tens of molecules to a million molecules. You can see that even across this large range of molecule abundance, there is still clear separation and distribution of dots in each of these 2 images that we are able to provide counts for. It doesn't all just blend to one big blob when you get to higher counts. Another really cool feature of our product is that because it's not microfluidic based and it's a spin column, it's possible to process so much more DNA as compared to other legacy platforms. Here, I'm showing you our throughput per reaction compared to other dPCR systems, where with those, when thinking about what actually gets analyzed, the overall volumes end up being really, really low. Because Countable PCR delivers a non-microfluidic platform, it allows you to process a lot more DNA input than your standard digital PCR. And then that's also one aspect of how we are able to have such high sensitivity. You're broadening your search by like that much more, a significant amount per reaction. So in summary, I walked through how the platform works and what the platform delivers in terms of high sensitivity. And I just want to summarize how Countable PCR really is a quantum leap for rare molecule detection. So 1, it really pushes the limit of detection with the highest sensitivity available. So you have more approaches to capture the rarest events, as we discussed earlier. 2 is that all these counts are actually absolute quantification. So there's no Poisson error estimation or standard curve matching that you need to do. All the counts that you get are directly from the sample itself. 3, it's completely automated data analysis, which reduces the time wasted for the user to determine if the data that they're getting is valid due to things like user variability or bias or anything like that. We automate everything for you. And 4th is, with a huge dynamic range that we support, you can measure rare and abundant targets all together in one tube. So for more common events that you may be using for like comparison or benchmarking, if you're out of range, you don't have to go back and redilute your samples or split them, whether you're looking at like rare or abundant molecules. And then 5th is, you can multiplex easily. So that's something multiplexing isn't really something I went over really in depth in this presentation, but I would love to explain it more for people who are interested. So basically, we can tackle multiplexing assays and get your assays up and running really, really quickly with our system. And then finally, I would like to announce that Countable PCR will be available in June 2025. This platform doesn't include just the instrumentation, but also all the consumables and reagents that you need to get your experiments going. So it includes kits for spin column, the matrix generation PCR tubes, the PCR mix, and for those who are interested in like fast setup for multiple targets at once, we also have a universal multiplex kit available. On the system end, it's also an integrated instrument with automated software for 3D imaging and counting. So it's able to run 192 samples in a workday. And then, oh, for this, I just really want to emphasize that when I refer to 192 samples, it's not like we're splitting a sample across multiple wells. We're aiming to run every single sample that you have by itself in one single reaction. And then also every sample also supports up to 4 channels for multiplex detection. Um, and yeah, I think that's it. So I really appreciate your time attending this webinar today and joining us in learning about Countable PCR. I would like to extend an invitation to everybody here if you're curious to see how it works for your own projects. We're currently in an early access program phase, and I'd be so happy to talk to all of you about how we can work together to make Countable PCR work for you. Thank you so much again, and I'll be taking any questions from the audience if there are any. Yeah.
Hi, I'm back. Thank you so much, Jeanette, for that wonderful presentation. I think it was a really nice introduction to how Countable PCR helps rare molecule researchers. So we had quite a few questions come in as you were going through that. So I'm just going to get started. So our first question is, what's the lowest number of molecules you can count in the system?
So the lowest number of molecules that we count in our system. So though the system is capable of picking out single molecules, and theoretically you can count single digits of molecules, there is always still that question of like statistical confidence. So that's an example of why we tested the pre-amplification protocol, and that's why I showed it earlier. So in the non-amplified samples, we had on average 6 targets per tube, which means an error of almost like 20% if we get like 5 versus 6 targets. But then when you're using like the pre-amplification protocol that I showed, we were able to up like both the numerator and the denominator to really improve that statistical confidence in the percent VAF.
Yeah. And next question. This is about the imaging. How do you ensure you don't miss a count if 2 signals are very close together?
How do you ensure that you don't miss a count if 2 signals are very close together? So that's a really good question because I did only show the image in a 2D space and as one image, but how the technology works is that it is a 3D counting tool. So every partition signal is imaged multiple times. And then when you look at the signal coming from a 3D space, that increases the resolution for you to be able to see whether the signal is, for example, 1 or 2 molecules. So in that sense, we're really counting the number of like 3D shapes that we're seeing and not just like 2D space and overlapping. That makes sense.
Yeah. I think also it's worth kind of following up with the algorithm that you mentioned, which also helps really resolve those 3-dimensional shapes. Next question. What is the minimum and maximum amount of DNA that you can load?
Minimum and maximum amount of DNA that you can load. So the data that I showed earlier had 3 micrograms of DNA loaded. And then the lowest amount that we've tested in-house was 1 nanogram DNA.
Straightforward question. How many colored channels?
Yeah, great. Good question. So every instrument has 4 color channels by default. So they're the standard red, orange, green, and blue. And then they work with our universal multiplexing probes as well as with many of the TACMAN probe wavelengths.
Kind of on the error rate, what is the typical percent CV that you would expect to see?
Typical percent that you would expect to see. So on a great assay, maybe like 5% or less, but it can range from like 10% to 15%. And at the absolute worst, like maybe 20%. But again, it's really based on your assay. So maybe like 20% may be fine, or maybe 5% is the max for you. You're in charge of the experiment, so you can really set like what is acceptable for you. But like, yeah,
Yeah.
there's a range there.
Yeah, and I think that also goes back to the sort of statistical confidence. Like, are you looking at the differences between 2 things versus like the absolute count of one specific target? So, Another question, how long is the actual time for sample preparation and reading time? So from start to finish, I guess kind of looking back at the workflow you showed, how long does that take?
Yeah. So it's on how you set up the PCR reaction. So it's a similar amount of time from when you set up a normal PCR reaction. As I alluded to in the slides, you can process and centrifuge 48 samples per spin in 20 minutes. So that's 20 minutes for 48 samples. And then the thermocycler that we use is just like a standard, like off the shelf 96 well thermocycler. So it's possible to amplify 96 samples all at once in the thermocycler, usually with a 1 hour protocol. So that's about so far 1.5 hours of time. And then that's all the way to the spin protocol. And then for the counting protocol on the instrument, we image and count roughly at the same time. So the final data, the first final data comes out within 5 to 6 minutes, I want to say, after you put the sample in. So the total time really depends on how many samples you're imaging at once. If you're imaging just one sample, you'll get it in like 5 to 6 minutes. But if you go through like 96 samples on all 4 channels, then it takes about like four, 5 to 6 hours. Yeah. So yeah, 5 to 6 hours for the imaging, and then like about 1.5 hours for the setup and amplification.
Yeah, thank you. A few more questions we have time for. Are you able to detect multiplex targets for rare molecule detection? So you showed this for one target rare and one target high, but what if both targets are rare?
What if both targets are rare? So yeah, we can multiplex rare targets. So in theory, it should not be a problem. The only thing to keep in mind is that for when both your targets are rare, you probably would want to run a positive control for each of those in order to really train our detection algorithm to be able to be sensitive for these rare targets. But yeah, in theory, it should not be a problem. You should be able to do that.
Yeah. You say there is no sample lost in countable PCR since you don't use microfluidics. But what about during imaging? Do you image and analyze 100% of the tube?
Um, oh yeah, so it's true that no template material is actually lost in the partitioning and PCR, unlike other systems. So in this sense, 100% of the available target material is given the opportunity to be detected. In terms of imaging, it's analogous to like tissue staining and characterization. So we're looking at enough of it in order to make a confident call on abundance.
Yeah. So looking at the time, I want to be kind of cognizant of everybody's day. So we'll wrap up now. There's still some more questions. If you ask something that didn't get addressed, we will follow up with you directly to get that answered. In the next few hours, you're going to get a follow-up email in your inbox, and it will contain more information about joining the sample workup program, including a calendar you can use to directly schedule a meeting with Eileen Chum, the founding scientist and our VP of Product at Countable Labs. And I want to thank Jeanette again for the great presentation and for sharing with us how Countable PCR really elevates the PCR game for rare molecule detection. And one last thank you to all of you for joining us today.